I'd just like to welcome everybody to this workshop session. So here we're going to be focusing on quantitative ChiP-seq analysis, and guiding you through this is Rory Stark, who we heard speak yesterday in one of the shorter talk sessions. I've known Rory for a really long time, as we both acknowledged in the green room before this session. And he's going to be an excellent hand in teaching you guys how to use some of the bioconductor tools for doing this kind of work. So take it away, Rory.
So if people have questions as we go, I'm happy to be interactive, and often it's best to stop right at the moment in the workshop when you have a question so that we can poke around a bit and maybe show you some other things. So if you have a question, what should they do? Raise a hand or write it into the...
So I guess to start with, if people put questions in the Q&A session, and Michael and I will try and keep an eye on that, and if something pops in that Rory doesn't see straight away, we'll point that out to him, and hopefully he can address the question straight away there and then. And if not, if it doesn't answer your question for whatever reason, raise your hand and we will essentially pass the microphone to you, and you can ask it out loud.
So for those of you who want to follow along, I don't know how many of you have done this, you can go to the workshop page that's on the main website page, and you end up, if you want to load it in the Docker instance that's being provided by the conference, you go to this link right here, this cancer data sci, and you have to kind of, I think you might have to actually register for the site and then find the workshop, just put in CHIP and you'll see it, and then it'll spin up an instance for you. The other way you can do it is to come, this is the workshop, there's the GitHub, but you can also pull the Docker image. And this will be up long after, it'll stay up. So you can run this workshop or in an environment where you have all the data and the workshop right in there at any point. And you can pull the Docker image. It's kind of large for some reason. I will say that the instances being provided by the conference are very computationally underpowered for what we're doing. So I've provided in the workshop the ability to skip the computationally intensive stuff and just load the result to keep moving on. But I'm running it on my own machine here where I've got 80 cores. And the interesting thing is I can run all the code here in under 2 minutes, but in the workshop instance, it takes over 20. So there's a big difference. Okay, so I'm going to get rid of that and introduce the workshop. So if you have an RStudio instance, I'm going to sort of assume you do. You're going to come in, it's going to look like this. And the first thing we want to do is load the workshop library, Quantitative ChiP-seq workshop. It's going to load diffbind and all of its dependencies, which takes a minute. It's probably shorter on mine than on some of yours. And there we are. And there's a little note at the bottom. There's a bunch of different ways of loading the workshop. If you say vignette, if you do that one, it will open it in the help pane. Matter of fact, I'll do it. You can open it in the help pane. That's not actually how I'm going to work today. There's some other options. You can open it in another window, right, like that, by running this viewer. Or you can open the source in a source window and then run the chunks. And that's kind of the most interactive way where you can really run things. I've opened it in another window, and I'm going to kind of be going back and forth to the window and to the screen. Okay. Are people okay? We're going to start. So what we're talking about today is quantitative, specifically differential analysis of these related types of assays, ChiP-seq, ATAC-seq, and related. And the thing these have in common is that you do something to samples that generally have some cross-linked chromatin, or the chromatin's involved in some way, and we're trying to interrogate not just the DNA, but the proteins around the DNA, the chromatin, the transcription factors, or we're looking for things like open chromatin, and then there's other assays. And we end up basically chopping it up into a bunch of pieces, purifying it, sequencing it, and then basically counting it. And so it's a counting assay, and that's really where we're going to come in, is really doing the counting and comparing. I'm going to do a little plug here. A few years ago, James Hadfield and I wrote a book chapter in this book, Field Guidelines for Genetic Experimental Designs. The name of the chapter is Characterization of DNA-Protein Interactions. It's about a 30-page book chapter. And if you send me a message, if you either email me or send me a message, I can send you a PDF of the whole book chapter. I can't just make it public, but I can send it to anybody who asks me. So feel free to ask me one way or another, and I'll send you a PDF. So instead of explaining all the stuff that happens before our analysis, I'll refer to the book chapter. I'm going to put up one picture, and this is kind of the picture that I use a lot, which is all the steps from after the experiment is run. And of course, the most important step isn't on here. It's in the chapter, I assure you, which is good design. You can't get a good analysis unless the experiment was properly designed. And the experiments we're going to be talking about today, you're going to be comparing two conditions, at least two. You can compare lots of conditions. You can do multi-factor analyses, but the minimal sense, you have to have a base condition and something you're comparing to and looking for changes. And you must have replicates. Replicates are absolutely essential in all of these enrichment assays, even more so, not less so, than more so than RNA. For some reason, people seem happy to do replicates for RNA, but they're a little skeptical on ChiP. And I understand some of those reasons. The experiments are often more expensive, particularly if your antibody is somewhat exotic. And the technical requirements of running the experiment are a little more difficult, onerous. So basically the cost of replicates is higher. But what we know is that there's more variance, technical variance, in running a ChiP or a ATAC than there is in running RNA-seq experiments. If you take the exact same sample and a very homogeneous sample like a cell line and run the exact same RNA-seq in multiple replicates, the variance is pretty well controlled, especially if it's the same person doing it. But you can have the same person with the same antibodies doing ChiPs on the same tissue, and there's a lot more variance. And the whole point of replicates is to capture the variance. So if we don't have replicates, we can't capture variance. If we can't capture variance, we can't get confidence statistics that we can be confident in. So that's my big pitch. There's a number of steps that you have to do just to get to the point where you can read this into Bioconductor. And the main one, of course, would be to run the experiment, to do the sequencing, to get the sequencing, and to do some sort of alignment. So basically, we're relying on aligned reads. There's a whole bunch of quality control that one can do to see if your experiment worked. I'm not going to cover that in this workshop. I'm going to point you at, there's a number of tools. We have one called ChiP-seq, which is a package in Bioconductor, which is actually related to diffbond. And that's this top stuff here. Okay, so you're going to do some kind of filtering and quality. Now the next issue, the big issue in doing a differential analysis is there's a bunch of tools, there's a lot of styles, but within the Bioconductor world, I would say there's really two, as evidenced by publications in the last 18 months, that is published analyses, there's two tools that are used the most. diffbind, which is the package I'm associated with and I'll be talking about today. And the other one is Seesaw, which was written by Aaron Lunn and the group in Melbourne. And the big difference between those two packages, they both end up fitting negative binomial distribution and using generalized linear models. They're basically, at the end of the day, they're doing the same thing. But the big issue is whether there's a peak calling step. Diffbind basically is relying on you having called peaks, and CSA is using windows across the genome, overlapping windows, and doing a more unbiased interrogation. There are advantages and disadvantages to each of these two. I'm going to be talking about having peak calling. There are actually some advantages of having the peak calls, both for quality control at the beginning and also for interpretation at the end. So for most people, the hardest part about doing the analysis is actually setting up the experimental data in a sample sheet. So people spend, getting all your data in the right place where it's all organized and you've got all the metadata captured in one place. Once that's done, you can actually run an analysis in one step. But if you haven't, then you won't be able to. So the first thing I'm going to start with is a sample sheet, and this is the sample sheet we're going to be working with a data set, the same one that I talked about in the talk yesterday. And what we're testing here is, this is a breast cancer data set. These are all breast cancer cell lines. There are 4 breast cancer cell lines, BT474, MCF7, T47D, and ZR75. We're chipping the estrogen receptor, which we know to be crucial in estrogen-positive, ER-positive breast cancers. And we're testing how the regulatory elements change in breast cancers that are resistant versus responsive to the drug tamoxifen. So tamoxifen is a drug that's used to treat ER-positive breast cancer, and it works until it doesn't work. We're trying to understand the mechanisms of tamoxifen resistance. So we have a number of cell lines that respond to tamoxifen and a number that don't respond. And crucially, because this is going to come, we have one cell line, MCF7, which normally responds to tamoxifen, but we also have created a resistant cell line, a resistant version of MCF7, which basically we just flood it with tamoxifen until you're left with resistant cells. And that way we can actually look at the transition. But there's a set of samples that are on both sides of our comparison that have something in common. We've done them all in replicate. And then what do we have for each of, so we have 11 samples. For each one we have a BAM file with aligned reads. Okay? And then we have, now this is a CHIP experiment, and we are using an input control. So we have one input for each cell line, actually each type of cell line. So we have 5 inputs. You notice this is using the same input. So an input is a CHIP where we just don't introduce the antibody. There's other types of controls. For an ATAC, usually you don't have this type of control. So there's a point when we'll talk about controls that you can do the analysis with and without controls. And the question of how to use controls is a big question. And there's different ways of using them. One of them is actually in calling peaks. It's actually one of the advantages of calling peaks for a CHIP is that you can use the control to identify enrichment and avoid enriched areas that are also enriched in the control. You're looking for enrichment over control. In our case, we're calling peaks, so we also have a set of peaks for each one. So this would have to be done outside. You'd have to run an aligner and a peak caller. In our case, we're using the max peak caller, although the format of the peaks is in bed format. So I'm just calling it a standard bed, chromosome start and name score. But we could actually put in the peaks that come straight out of max and put in max or narrow peaks. There's a bunch of different options here. So step 1 is getting your sample sheet ready. Okay, so now let's come back to the analysis and see what we can do. The first thing we have to do is be in the right directory where our data are. And if I come here to files, this is actually up here is the workshop package, and the data are in inst ext data. And you see, that's our sample sheet that I've got up in the spreadsheet. That's where the read files are. That's where the peak files are. And so it's relative to where the sample sheet is. You can see these are relative paths, right? So if I look in reads, I can see the BAM files. I also have index files. Oop, that was a mistake. Sorry about that, I just dropped out, but I should be able to jump right back where I was. Yeah, I'm right back where I was. Okay, so we're going to set our working directory to inst data so that we're in the right place. Down here, hopefully you can see that. If you have any issues, flag them up, and I can change my style or make things bigger or something if we can't see things. So we can see from here we're in the right place. Peaks, reads, and the plots .csv. We come here to plot, make that a plot. So this part of the workshop here is everything I just talked about. The sample metadata. In our case, we know about the interesting metadata is the tissue, the condition, and the replicate. Now, everything else is, that's the same for everything. That's the same for everything. So condition and the replicate, and called peaks. So we can read in our sample sheet and just have a look at it. I'm just reading that in as a CSV. You don't have to read in the sample sheet to do an analysis. I'm going to move this over because I know this takes a lot of space. Here we can just see it as a data frame. That looks just like what we saw in the spreadsheet. Now, once you have these loaded, you could actually, within diffbind, run an entire analysis with this line. Don't do this right now, because this would go do every step. So it's a default analysis. One of the things that diffbind does is it attempts at every step to have sensible default. So if you just give it a minimum of parameters, it's going to do something that it thinks is sensible, meaning I think is sensible. But there's usually a lot of parameters where you can get fine-grained control. So if you run just DBA analyze with a sample sheet, it basically runs these steps. It loads the sample sheet, it applies a blacklist and generates gray lists, it counts the reads, it normalizes the data, it sets up a model and contrast, and it runs an analysis. We're going to go step through all of those one by one, as opposed to if you just run this like that, it will just run all of these automatically. So the first thing we want to do is actually load the sample sheet as a diffbind object. So that's down there. So you see it's loading the 11 samples that were in our sample sheet. Kind of gives us a little feedback as we're doing that. And now we have an object called tam.peaks in this case, and we can examine that object. And here's what we see. It says there's 11 samples, and then it says 2,845 sites in the matrix, 3,795 total. And I'm going to explain what that is because that's pretty important to understand. But for each one, we see the metadata, and what's been added here is the number of peaks that were identified. So that was identified externally. We haven't done anything. That was identified by max, in this case, the max peak column. So you see each of these has a certain number of peaks. In order to build the binding matrix, the peaks are merged. So any peaks that overlap are merged into a peak that in an interval that's wide enough to encompass all of the merged peaks. So the more samples you have, actually the wider and wider your peaks get. But we end up with unique, non-overlapping peaks. And in our case, we have 3,795 of them. We've defined a default consensus peak set of 2845, and that is what I'm going to talk about next. Actually, no, what I'm going to talk about next is we're going to do some filtering of these peaks, and then we'll talk about how we come up with our consensus. So the main filtering we want to do is we want to apply blacklists and gray lists. A blacklist are regions that have been identified, and this has been come out of the ENCODE project, very large project looking at these regulatory elements of known problematic areas, places where parts of the genome where we know that we don't get reliable data. And the idea is just to filter them out. And that has proven to be best practice. Over time, the blacklists have gotten actually smaller and smaller as we've resolved different parts of the reference genome. So diffbind has a function called DBA blacklist, and I'm going to do this for a bunch of these things. I'm going to pull up the help page, show you what DBA blacklist looks like. It's in there for some reason. So DBA blacklist really does two things. It makes a blacklist, and it also makes something we call a gray list, which I'm going to talk about. And it lets you retrieve them so you can see what's been filtered out. Or in the case of gray lists, which are computationally expensive to make, it lets you save them for later and control some of the parallel operation here. So let's do a blacklist first. Usually we do them both at once, but the blacklist we can all run, even in the conference Docker. And what happens if we run the, hello. So right now I'm saying ignore the gray list, just run a blacklist. So what diffbind does is it automatically detects what reference genome was used. It goes into the BAM file, and if there is a blacklist for it, and there's only 5 or 6 that have blacklists. Actually, these are the ones that we have known blacklists for. It retrieves the blacklist and filters out the intervals. So basically, it's saying, of all of these, 3 peaks were eliminated, and those 3 peaks overlapped each other. Only 1 consensus peak. So they were from 3 different samples, but they were pretty much the same region. So we've basically eliminated 1 peak, and now we've got 1 fewer. So that was it. Now, what's a gray list? A gray list. The concept of the gray list is that in each individual experiment, if we run controls, this only applies if you have controls, so it tends to be more in CHIP than in ATAC. We can look at the controls for anomalous regions, that is, places where there's unexplained pile-up of reads. So if there's a big pile-up in your control, you really can't trust a peak called in that region. That make sense? So my co-author wrote a package called gray list CHIP. See if it can. If that's there. Gray list CHIP. I think it's because he doesn't have a. Oops. Oh no. Okay. I can't remember how it's called, because oh, here it is, gray list objects. And this is the package, and if I come here to the index, this is his package. I'm having a hard time with that. Okay. And what gray list CHIP does is you give it control, and it looks for anomalous regions, and you can control some of the cutoffs. So by default, it will attempt to build a gray list if you've specified controls. Now, this is actually a computationally expensive task. It has to look at all of the reads and all of the controls. It has to build a model of them, and it has to exclude outlying regions. So we're not actually going to run it. I can actually run it. It doesn't take that long. I could have run it while I was talking, actually. But you can see what it does is it counts the control reads, and then it builds a gray list for each of the input controls. And you can see that it's excluding 0.21%, 0.14%, 0.07. So that's typically what's happening. You're excluding some very small fraction of the genome. But what we found is these peaks actually do show up a lot in results, and they really have changed some of the biological conclusions once you apply this filter. So what I've done in the diffbind package, I've included the gray list for the sample data. And get that so it's easier. And so there's this object called tamoxifen gray list. And if we look at it, we can get the names. And it's, I think it's a list of G-range lists, I think, actually. And there's a master list, and then there's one for each control. And the master list, let's just have a look here. Gray list. So this is the gray list for the experiment. There's 72 regions that it's identified as being anomalous in any of the controls. This is a union of all the controls. So if it's no good in one control, if it's no good in one of your samples, it excludes it for all of them. There's different ways you can control that. And indeed, if we look at the controls where there's an individual one for each control. There's length 5. And if I do the names, you can see that there's one for each control. So there's the one for VT474. So you can save these. Technically, it would be better practice to compute the gray lists and then filter the reads, the alignments, before you call peaks. Because something like Max builds a model of the background, and it's building a model with these anomalous regions. And if you remove the anomalous regions, you'll actually get a slightly different set of peaks. So you can go back and recall peaks. That would be ideal, but we found that pretty much you can eliminate them at this point. So what we're going to do now is eliminate the gray list controls as well. So I'm going to say, I'm going to give it the gray list that we pre-computed. If you had just said gray list true, it would have computed it for you. In this case, it's applying the blacklist again. Okay, so now we see those are those 72 ranges. 72 ranges. It eliminated, there was 420 across all 11 samples. And those 420 peaks actually merged into 50 peaks. So there were 50 merged, actually there were 52 merged peaks, of which 50 appeared in more than one sample, which I'm going to talk about next. Okay, so now we've filtered our peaks. I'm going to stop there for a sec. Does anyone have any questions? No one's indicating any questions. I don't see how to see them, so that means if I click on direct messages. Nope. Okay. Session. Okay. So now the next step is forming a consensus peak set. Ultimately, we need a matrix of the peaks we're going to consider, whether they're differential, and look, we're about a third of the way through, and compare them and count all the overlapping reads. So the next step, so the matrix that we're going to do our analysis on, the rows are consensus intervals. They could be peaks, they could be open chromatin, whatever the histone marks. I tend to use the word peaks, but sometimes I use intervals, sometimes I use regions. But when I say peaks, it doesn't have to be a peak, right? It's just some genomic region. It's something you would have in the G ranges. So the rows are intervals, and the columns will have 11 columns, one for each of our samples. And then each cell will have a count. That's what we need to get to in order to use something like DE-seq or EDGE-R to do a differential analysis. So there's a lot of ways of forming consensus. We can talk about that if people had questions. The easiest way is just kind of to get peaks that overlap. And there's a
so there is actually a question now, Rory.
Oh, I see QA it just showed up. Are there tools for filtering? I probably need a little more detail of what type of filtering. If you want to type in, this is Anna, right? Anna, if you want to tell me what type of filtering you're interested in, while you're doing that, I'll tell you what filtering there is. There's blacklist filtering, there's graylist filtering, there's filtering reads by quality, by duplicates. But I'm not sure if there's other filtering. Okay. Again, if you want, oh, there it is. Right, so I got the question, are there tools for filtering, but which filtering what? The blacklists and the gray list are inside the package, right? So I just showed that filtering. Well, the other thing that there is, and I'll show you this when we do counting, is we filter out peaks that don't have enough reads, just like you would filter out genes when you're doing an RNA-seq that don't have enough reads. There's a number of types of filters, and actually the book chapter talks about them. Again, if you want to be more specific, I can address something more specific.
Rory, could it be that what Anna is asking is how would you do what you mentioned? So how would you go back, remove the reads that fall into these problematic regions?
No, that's outside the package. Right. You would need to do, I'm trying to remember what we use, SAM tools. There's ways of taking a BAM file and literally removing reads from it. And that's what we do. By the way, we do that in our institute. We actually do take them out of the BAM file, so they're not even seen anymore. The blacklisted reads, but we've got it in a pipeline. So if you're just sitting there at the command line, it's kind of a pain. I can't remember what tool we used. Okay, that's a good clarification. So let's look at overlapping for a moment. There's this function dba overlap. And what this returns with this mode overlap rate is a vector. And you'll notice that the vector is 11. Okay. And what this is showing is this is how many peaks there are, that merged peaks that are in at least one sample. This is how many peaks that are in at least 2 samples. So it's been identified twice. This is how many were identified in at least 3 samples, all the way to the ones that were in all 11 samples. There's only 119, right? So this is our universe here of 3743. And so one of the easiest ways to make a consensus is just pick one of these numbers. And one of the nice ways to do that is to plot it. It's actually worth, this is a QC plot as well. We can see that plot, right? So this is how many samples it overlaps, and this is the number, and this is just these numbers plotted. What is roughly a geometric drop-off, and this is because peak calling is actually quite noisy. So if each level is half of the previous one, if it's no worse than that, I feel okay about the experiment. If there's a drop-off worse than that, often you have some outlier samples that might need to be eliminated because they have nothing in common with anything else. Here you see we're actually doing better than geometric. It's better than half for each of these. It's a pretty good experiment. We get pretty high agreement. We've got a pretty good antibody there for the estrogen receptor. So the question is, should you be lenient or should you be strict in what peaks you let into your analysis? And what we suggest is you should actually be pretty lenient, because the statistics that we're going to be using to identify differential peaks are quite robust. So even if you put in bad peaks, it's still, they shouldn't come out as being high confident differential. So we tend to use more on this side of the graph than on this side. We could use this, the universe. We found that if you just kind of go to 2, it's at least identified twice. Again, you should have several replicates. I'd say the minimum experiment you should be looking at would have 6 samples. That you're going to get a good universe of peaks and get a good result. And that's the default. And that's why when I look at CAM peaks, I'm getting 2795 as the default, right? So this 3743 is all of them, and this 2795 is that. And this is what we call the overlap rate. So we say it's the overlap rate of 2. There are more advanced ways of doing this. For instance, we could come up with a separate consensus for each of our cell types and then merge those. Say, well, I want it to be in 2 out of 3 of my replicates and then take the union, things like that. And these are in the diff-bind vignette. We go through some of the other ways of more advanced ways of generating consensus. The other thing is you might have a set of consensus peaks that you know externally, or regions of interest. Promoters. Maybe you're looking at H3K4 trimethyl or something, and you just want to look in promoters. Then you could just feed in the list of promoters, and you don't actually need to call peaks at all. So DiffBind you can run without having peaks called for each sample. And we can see the consensus peak set. We can retrieve it with the DBA peak set thing. So I'm just saying retrieve this peak set, and then get that back as a G ranges. All of these are scores, so we can sort of see, for instance, peak 2794 was identified. If it's not 0, it was identified as a peak. That it wasn't identified in T4 7D, but it was identified in all of the other sites. And there's overlap and Venn diagrams and things you can use to have a look at this. We can go into that in more detail if people like. So at this point, we're going to use the default consensus peak set and counting leads. Okay, so this is another computationally expensive task that won't really run very well in the instance that you have with the let me look here. With the conference. But in general, you would call DBA count. So don't run this. I've got the counts pre-calculated. But let's have a look at DBA count and see the kinds of things that you can control. This has a lot of options, as you can see, DBA count. So you give it a DBA object. You can give it a peak set. For instance, if you had the promoters, if you leave that empty, it'll attempt to create one by overlapping the peaks, and there's your default of overlap. There's a score, which is used for plots. There's a fragment size, if you know it. I'm going to talk a little bit more about what DBA count does in a minute. And now there's a couple quite interesting ones. These parameters are actually quite important. I mentioned that when we merge peaks or intervals, whatever they are, they end up getting wider and wider and wider. And this causes two problems. One is you end up with a potentially large variance in the size of each interval. And this can actually throw off the analysis. Obviously, we're using tools that do transcriptomic analysis and genic analysis, and those are quite different in size. So we do control for that and adjust for it. But there's certain types of plots and things where it becomes confusing if you have lots of different sizes. Particularly if you're doing experiments where you expect them to be roughly the same size. It's a binding site. It's open chromatin, which actually, kind of has a maximum, particularly if you're doing an ATAC-specific experiment where you know the ranges, there's specific ranges of widths. The other thing is, if they get too wide, what happens is for most of the samples, for most of the peaks, most of the reads are actually not your signal, they're background reads. Because the signal is actually concentrated in the center, or quote unquote, the center of that region. And you've kind of captured all the noise of the peak culling, and you're injecting a lot of background reads into the analysis, and it can throw off at the margins what is identified as being differential. You're actually much better off taking a sample that's highly enriched from those regions. People get very, I think, over-obsessed with getting the exact margin of where their peak is. And if you actually look at what you're doing, what kind of biological questions you're asking, that's rarely important to the biological question. I'm not going to say never, but usually you just want to know, is there enrichment near this gene? Is there enrichment in this enhancer? Have I changed my chromatin, in this area of the genome? And actually having the exact boundary isn't important. If you can get an area is enriched in the center of that, and be comparing those across all of your samples, you're going to get a better signal for what's differential and guide you to the right parts of the genome. That's our concept. So we do something called recentering with summits. So the counting is a two-step process. In the first step, I'm actually going to run the DBA count because I'm talking. I may as well run it. And on my machine, it'll run fine. There we are. So computing summits is what we're doing now. It's a two-step process. So one is it counts. It takes the consensus peaks. It counts all the overlapping reads in all of the samples, whether or not the peak was called in that sample. Right? We're going to still look to see if there was enrichment, even if it wasn't called as a peak. And then we look at the pile-ups across the merged peak, and we find a consensus center point, point of highest pile-up. Then we rebalance, basically recenter the peak. If I look here using this value, which is 200. So I take the point of the highest pileup, and in this case, 200 base pairs upstream and 200 base pairs downstream. So all of our peaks are 401 base pairs. So even if they could have gotten very big, but we're taking a 401 base pair. Now that number 200, 200 up, 200 down, plus the center is how we get to 401. There's different numbers you might want to use. For ATAC, I usually use 100. Sometimes I've used 50. For wide histone enrichment, I've used 500. So this is kind of optimized for the kind of reads we do for transcription factors. So there's a little bit about that in the vignette. So once the peaks are recentered, now we have a new set of consensus peaks. We have to go back and recount. So actually we have to make two passes through all the BAM files, and that's what you see here. Compute the summits, recenter the peaks. One of the new features in DiffBind is it will actually detect whether you're using single-end or paired-end sequencing. There is a limitation where it will apply the same for the whole experiment. So if you have mixed single-end and paired-end, you're probably best off treating it as single-end. It only counts one end. And it'll respect the paired-end and single-end. The other things I want to point out are filtering. So this is where we filter out peaks that don't have enough reads. This is an RPKM value. So we're filtering out all the consensus peaks where there wasn't at least one sample with an RPKM of 1. So that's normalized for the library depth and the width. Generally, we keep duplicates in. I've actually done a whole bunch of work recently on duplicates in general. Well, for single-end sequencing, you should definitely keep the duplicates in. If you have paired end, you might want to remove them. But in general, duplicate reads, duplicate alignments in DNA enrichment assays are more likely to be true biological signal because we get very high pileups in very narrow regions. So there's got to be duplication. And then there's an issue of what to do with the control, and I'll come to that. Okay, I'm going to take a pause there for a moment. I'm going to take a pause a moment. So now we have a counted, we have a new object here with the counts in it. I just want to stop if there's any questions. Yeah, we're about halfway through. That's pretty good. Look here. Just want to see if there's any questions. No. Okay. In our new object, cam counts, what do we see? We see 11 samples, 2795. So now, before, remember in cam.peaks, every sample had a different number of peaks associated with it. But now in cam.counts, they all have exactly the same 2795. That's our consensus set. Was it actually? I want to see if it was 27. Yeah, it was 2795, so nothing actually got filtered out. So I'm gonna delete. So this is what we're looking at here. Oops, that's what we're looking at right there, the highlighted stuff. So what do we see now? We have the same metadata that we had before, but we have two new columns. we have the number of reads. That's the total number of aligned reads in the BAM file. So that's the sequencing depth. And then we have this guy, FRIP, which is the fraction of reads in peaks. And this is when we start, now we're going to start talking about normalization. This becomes very important. So this is the proportion of these reads that overlapped one of these consensus regions. And you can see the numbers range from, I think, a low of 0.06 to a high of, what, 0.32, right? Now, what accounts for these numbers, and what numbers do you expect? If you go look at ChiP-QC, there's some QC stuff to look at what you should expect for enrichment. And we compare it to a control. In the control, you should have very low enrichment. But in a CHIP or an ATAC, you can get numbers. They should be above 0.1. I would say that's not a fantastic replicate, but that's okay. That's why we do replicates. And it can go up to 0.5, 0.6, particularly in ATAC. I think we see higher numbers. But there is a lot of variance, and what accounts for this are a couple things, but mostly it's the CHIP efficiency. People have in their mind in these assays that you pull down just the enriched stuff, but all you're doing is enriching. So if before you do the pull down, the signal you were interested in counted for 0.01%, and you end up with 10%, that's a huge amount of enrichment. But it's still 90% of your reads are background reads, non-signal reads. That's actually to be expected. But it does create an interesting issue when you think about normalizing. So we have quite a bit of variance in the depth of sequencing and quite a bit of variance in the efficiency of the enrichment assay, as evidenced by the fraction of reads and peaks. Is that clear? So just have a look again. Okay. So what's our next step here? Some plotting. We have the binding matrix. We have basically our raw count. And by the way, you can retrieve this if you want. Binding matrix, you could write it out to a file. You can get it out as a G-ranges object. I'll show you how to do that. These are actually normalized already. So there's a default normalization, just like we had the default consensus. Like I said, diffbine's always trying to do something sensible. But so here you can see, if I retrieve the binding matrix, I get a G-ranges object with the 2795 ranges, and a count for each, a normalized count. You can tell they're normalized because they're not integer each other. You can also get it back as the raw counts by changing the score. If someone's interested, I can show you that, but I'm gonna move on instead of doing that. So now we want, so that's the first step in looking at it. But the next thing we typically do is kind of try and get, at this point, we can get a nice view of our experiment, particularly looking for clustering, right? Clustering and correlation. And the easiest thing to look for is to just do the default plot. And we're going to look at the default plot. So I just say plot, and counts, and I'm going to pop this out so we can look at it in a little more detail. So the default plot is a correlation heat map. So basically what we've done is taken each of these columns of scores and correlated them with every other column, right? So we get an 11 by 11 correlation matrix and then plotted this heat map and see how it clusters. So these are just normalized counts. We haven't identified anything differential yet, but sometimes it's useful to see, are there outliers? Are there batch effects? Do your data naturally cluster by the biological condition of interest or not? And what we see here, we see there's, oops, sorry about that. There's a couple clusters here. There's this cluster down here, which is all of the MCF7 based samples, right? So remember, some of these are responsive to tamoxifen, and some are resistant. But the resistant and responsive versions of MCM7 are still more like each other as far as their ER binding profile, their estrogen receptor binding profile, than they are like the other samples. So that's something we know we're going to have to model. We're going to have to deal with that in order to get a result, because these guys are going to want to cluster together no matter what we do, even though the thing we're most interested in is what's different about them. And then the BT474, which looks like it's resistant clusters with that. But we could also see that the replicates cluster really well. That tells us there's no really bad replicate, right? This is one of the reasons we do replication, right? These are the ZR75s, these are the T47Ds, these are the BT474s, these are the resistant MCF7s, and these are the responsive MCF7s. Okay. And we can look at this another way. We can look at it as a PCA plot. Hopefully we're going to see something. Why did it not like my graphics state? Yeah, here it is. That's the same plot on both sides. So here we've just plotted it, and by default it's plotting it by tissue. And we can see here these MCF7s on the first component are all off on one side, and these are the replicates. So nothing super suspicious here. If we wanted to look at this by condition, that's the biological condition we're interested in. We can see they don't particularly cluster naturally in a PCA by the resistive versus responsive. They're not separable. There's no single line you can draw here in any of these components. And the other thing you often want to look at is replicate. Just look for a batch effect. We know that things are going to come out pretty good. There's no clear replicate that's jumping out. You can put labels on these, by the way. I think in the example there's one. What did I put here? There we go. Oh, that was with to label the tissue. That last one I can add a label. I just added this label, and now we can see that we've annotated the dots with what tissue they came from. Okay, these are obviously useful plots, PCAs, good stuff. And that kind of gives us some sense of moving to the next thing, which is normalization. So I'm going to talk about normalization in two phases.
There is a question, Rory. Sorry to interrupt.
Yeah, I should click to that. There we are. Hello. Where's the question?
I can read it to you.
I don't have a new one.
It's from Thomas Nake, and he's asking, hi, are there any ChiP-seq specific, compared to other technologies, metrics to test for a batch effect besides visually checking for batch effects? Are these implemented in the package?
Well, what I should show, I mean, what we because we actually do it programmatically, looking for outliers. And if I look at this plot, and by the way, the default plot is actually a heat map, right? So that's the default plot. It returns a correlation matrix. Actually, it actually returns. Why am I getting this? I think it just doesn't like. Oh yeah. We get back the correlation scores, and we check the distribution of correlation scores so that the replicate should be more highly correlated than non-replicates. That's it. It's a very simple check, but it usually flags something in a pipeline context. No, obviously we generate reports and look at them as well. The ChiP-QC package has some other plots that compare replicates and compute some statistics. There's the cross correlation statistic to see how well the whole enrichment profile, rather than just a single correlation measure, compares, a normalized comparison of that in the ChiP-QC package. Okay. That's a good time to ask a question because I'm going to move to normalization. So I was going to talk about normalization in 2 stages because it's a whole topic as we discovered yesterday. In the first one, it's sort of what's the default normalization? How do we even think about normalization or do it? There's another plot, which is the MA plot. And it's useful at this point to do an MA plot of our non-normalized data. Do I have it there? There we are. So hopefully people are familiar with MA plots. This is the x-axis has the mean concentration, that is the number of reads. So things that have a lot of enrichment are here, and things that have a little enrichment are here. And the y-axis gives us a fold change, in this case a log 2 fold change, between the way we're grouping our samples in these 2 conditions. The blue line is the center line, is this log fold change of 0, where there's basically on average the same. And then we've added this red low S fit that helps you kind of see the shape of your data. And usually what there is kind of down here, there's a center of gravity, a point of greatest concentration of your peaks. So every dot here, and this is smooth, but every dot is one of those 2,700 peaks. You'll notice that the center of gravity here is below the line. Below the line means that it's more enriched in the base condition, which is responsive, right? Resistant minus responsive, so it's negative, so it's more enriched in the responsive condition. So we observed that about our data. There's other plots we can do, for instance, in the vignette where you could see a trended bias. That's something that is technical, where the bias in fold change is correlated with the concentration. So as you get higher concentration, you get a higher fold change. And that's often indicative of a technical issue, although it can actually still be biological. The hard part, as I talked about yesterday, is knowing what's a technical bias and what's a biological result. So there's 2 ways of looking at this plot. You could say, I've got a bias towards responsive, maybe I should try and correct for that. And the other is to say, hey, I just made a biological discovery, which is the estrogen receptor loses binding in conditions of tamoxifen resistance. Maybe that's how tamoxifen adds to the resistance by stopping the estrogen receptor from binding. That is, the ER binding is what is interacting with tamoxifen, which making it work. So have I made a biological discovery, or have I screwed up my experiment? Does anyone know the answer in this case? No, really, I want to know. We did publish this many years ago, and I've changed my mind on it a few times. So it is good to see whether there's obvious biases or things that don't correspond to what you expect. So a lot of it is expectation. In RNA-seq, we expect there to be a core set of genes that don't change expression. And in general, we expect, if there are changes in expression, we expect them to be in both directions, gains and losses. But in chromatin and things like transcription factors, that's not the case. You could have a loss of binding in a way that you can't just have a loss of expression. If you have an experiment where one of your conditions loses all gene expression, it's dead. Right? There'd be a clear phenotype. It would, but you could have a much bigger change. And this is one of the key ways that these enrichment data are different than RNA data. The other big difference is in RNA, the range of expression values is generally higher, right? So even within one cell, you can have a transcript expressed at a very low level, a very high level with hundreds of thousands. While if you're looking at chromatin, well, let's say you've got two copies of the DNA. The most data points you can get is 2, right? It's not, you can't have 100,000 transcripts coming out of one cell. I mean, that's one of the reasons why single cell is difficult in this area, and we rely on the bulk. But these really are different data, and there's a temptation to just treat it like RNA-seq data. So I'm going to do the most simple normalization, which is, well, let's look at DBA normalize. This is a new function.
Okay. That looks good. Yep.
All right. Okay, we're back. We're looking at the normalized thing. I can't believe that 16 people that there's still 16 people who stuck with it. So that's wonderful. Thank you. We're going to look at this normalize function here. Hopefully, some of you did attend the talk yesterday, so some of this will make sense. But basically, when you normalize the matrix, and you normalize it specific to an underlying analysis method. So diffbind runs using either DE-seq2 or edge-R to fit the linear model and do the tests for the differential analysis. So you can normalize differently, and they actually handle normalization differently. You can't use the exact same normalization, although you can use the same methods. So you tell it what method you're going to use. You tell it what normalization, specific type of normalization, and then this library is where you set the reference reads, and that was what my talk was about yesterday. And then this is whether it's background or not, whether you're using background reads, whether you're using spike-ins, or whether you're using an offset matrix. So the default is to do a library size normalization. It's incredibly, it just divides by the library size. And let's have a look here. So if I say cam.counts equal dba.normalize to the default normalize. I'd be wrong. Well, I tell it cam.counts. It's not right. That should be instant. And then we can actually, I'm going to actually show you this. This isn't actually in the in the yet. So you can see what it's done is it's using the full library size and full and by library. These are the library sizes. That's what we saw in our object, and these are the normalization factors it's computed. So this is a big one, a larger one, and it gives you a larger one. If I'd done this for edge-R, you would actually see different numbers because it calculates it and uses it in a different way. So given that, we can see if we can plot what this looks like with the default normalization, and I'll put that up. It gets easier to plot MA plots once you've actually done a differential analysis because you just tell it the contrast and you don't have to give it too much information. And if we compare it to the previous one, I'm going to put them next to each other. This line's a bit closer, but we still have the bias towards responsive, which actually is what I think we want. Okay. So now we're ready to do modeling. We're actually ready to set our model, our design, establish our contrasts, and call the underlying analysis method to fit the negative binomial, fit a generalized linear model, and do testing. So the next key function is one called DBA contrast, which I just spelled wrong. Okay, so DBA contrast is where you set up your model. This is a big change in diffbind from previous versions to now. In the current version now, you can do any kind of model, any number of factors, and it kind of had a retrofit into the old one. So the arguments are a little confusing, but basically you establish a design and a contrast, and all the other ones you can basically ignore. So again, it always tries to do something sensible. So if I say dba.contrast cam.counts, I'm not going to save it. I'm just going to see what comes back. It's added this bit at the bottom. Sorry, see this bit at the bottom? That's now been added because we've now established. So we establish a design, and what it does is it looks for any case where there's some metadata, where there's different values, and there's at least 3 samples on 2 sides of that, and it tries to create a design and establish a contrast. And with this design, the only thing where we have 3 replicates at least on 2 sides is in the condition, where we have 4 resistant and 7 responsive. And so it establishes the default design of condition, tilde condition, and the single contrast of resistant versus responsive. But we can control that. Now I'm going to skip ahead because of time. I know from modeling this data, I remember we looked at the clustering, that we really need to take into account the fact that we have all this biological variation in our tissue types, particularly this issue with MCF7, but even all the other tissue types. And it would be nice to be able to model that as well. So what I can do here is, let's say I'm going to do the default one. I'm going to call this tam.model. And repeat that. We have the same thing, right? But all I'm going to do now is I'm going to change the design to a multi-factor design. And it's as simple as going tissue plus condition. So one of the things we can do here is see you see it keeps saying computing the results names. I can see basically what the columns in my design matrix are. It's created a design matrix, and I can establish any contrasts I like and as many contrasts I like based on this 1, 2, 3, 4, 5 column design matrix, which is implied by the. I actually haven't shown you this. You can see now at the bottom here the designs have been replaced by tissue plus condition. And that leads us to running an analysis. It turns out that DBA analyze is actually one of the simplest of the functions now. Because it can do everything, you have to give it some historical stuff. But basically, you just give it the method. I could change the design here as well. So if I say tam dot, I'm going to call it model equal DBA dot analyze. Model. So by default, it's going to run an edge-R analysis. I can actually run both. I kind of like to do a lot. Method equal DBA all. So it's going to run both an edge-R and a DE-seq analysis. So you'll notice it said normalize edge-R with defaults. That's because when we did our normalization originally, we only normalized for DE-seq. So as soon as it went to edge-R, it said, oh, it hasn't been normalized yet, so I'm going to use the defaults and add that in. It does this in parallel, by the way, so I believe the DE-seq one costs a little more. And now I can look at the results. So now it's added 2 more columns here, which tells us how many it's detected at differentially bound at the default false discovery rate of 0.05. And you can see they're pretty close. So now what we want to do is basically look at the results. Right? And the easiest way is to do that as a report. If you just need to get your let's say results, I'm going to call it report, DBA.report, model. So by default, it's going to use give us the DE-seq results, but I could change, for instance, the FDR. Actually, it's called threshold. To make it 0.01, I'll get back a few smaller, just to show you an idea of some of the things you can do here. And now I have a G-ranges object with 509. If I use the default, I'll do that. Default 0.05. We'll see the 783 that we saw here, right? So we're getting these 783. And it gives us a number of statistics for this. If I do that, is it going to be nicer? It gives us the range, concentration, so that's the log of the normalized read counts, and these are the concentrations in each of the two conditions. This is the fold change. It's the fold change that's computed by the ESeq. So you get kind of different results with the ESeq and EdgeR here because one does shrinkage. The p-value and the FDR, and it's sorted by FDR, and you see it goes right up to about 0.05. And so for this, obviously, you could do downstream analysis, you can annotate, you can do motif analysis. This is sort of the main way to get your results back, and you can write things directly to it. The other thing you can do is plot, and so I'm going to show you the plots. Obviously, we have our MA plot. It's as simple as saying, now that we have an analysis, and that's the 783. You could see if it was any different than EdgeR. Pretty similar. Yes, a little bit different, so you can start to compare results, see what it looks like. The other version of this is a volcano plot, which people, which can be useful as far as seeing the relationship between the concentration and the confidence, quote, significance. And yep, we can get volcano plots, and then then back. So you remember we did this. We plotted the counts, and we got this clustering where the MCF7s all clustered together. That's using all of the consensus sites. Actually, it would, by the way, if I put it via tam.model, I get exactly the same thing. But if I say using contrast equal 1, so there's one contrast, it's only going to use those 784 differential sites. And it'll change the clustering. Now, you might expect that we would get a perfect clustering, right? Because we only identified, we just did a statistical analysis to give us sites that were differentially bound between the two conditions. And you can see it's not quite perfect because here's the condition here, and this is also showing us the condition. The MCF7 are still close to each other. Probably easier to see in a PCA. Sometimes you can do something like reduce the threshold so that you get only the most significant sites. But part of the reason it's doing that is because we use the multi-factor analysis. So we're still using kind of the raw counts. They haven't been adjusted for the more complicated modeling we've done with the multi-factor analysis. So it's hard to see on that point. Let's see what a PCA looks like. Model contrast equal 1. Yeah, you can see that it's still that. Interestingly, if we do the single factor analysis, if we just do responsive versus resistant, it does actually cluster fine. But anyway, that's just different ways of having a look at these data. In fact, here in the vignette, I did a single analysis, and you can see that how it clusters. So in this one, I want to show you the model is there it is. The model's just conditioned. So it does show you that once you start getting into multi-factor models, you have to look at your data a little bit differently. We can also get a traditional heat map that is of the concentrations to show where things are gained and lost. And I skipped over the design. And the reason I'm rushing right now is in the last couple minutes, I wanted to talk about other types of normalizations. And these are kind of the plots that I had in my talk yesterday. And the workshop, this vignette will walk through how to do it. And the vignette that's with the package, the diffbind vignette, actually does it in even more detail. And in particular, it shows you how to do spike-ins and parallel factors. people, I think, have a good well, not a great understanding, but spike-ins usually where you use exogenous DNA from another species, typically Drosophila, some chromatin, from a chromatin pulled out. And if you can quantitate it perfectly at the experimental phase, you can normalize to that. But we've actually found that the spike-ins don't work all that well. It's hard to quantitate perfectly, and you don't capture the precipitation, or particularly the antibody efficiency, because there's a sonication step, and that can cause some of the bias. But the chromatin you're spiking in wasn't sonicated in the same way. So one of the other things that we've used, and there's a guy, Andrew Holding, in our well, he just went up to I don't remember where he went, but he's in our institute, did some analysis where if you have a factor that you know shouldn't change, and he used for this breast cancer stuff CTCF, you can chip for both of those, and then you know the CTCF shouldn't change and the ER should change, so we normalize to this. We call peaks separately and normalize to both of them. I want to take some. I've got a couple things to finish up, but I wanted to give one more opportunity to answer questions.
If I may, Rory, I have a quick question regarding these spike-ins.
Yeah.
There are these indexed chip protocols where you label, basically, you barcode your fragmented chromatin from different samples and then do a single IP with all the replicates and all the different conditions. Do you have experience with that? Does that work better?
You know, I've handled that once, and it was a good experiment. The problem is I personally have an N of 1, but it makes sense, right? Because you're doing it together. Particularly because we have demonstrated that the fragmentation step is a big impact on the differences between the libraries. Have you used it?
No, unfortunately not. No.
So the one I saw was good, but it could have just been good. I've noticed there are certain people who can come up. People say, "Oh, there's really high variance in CHIP," and then there's certain people who are very good at having low variance. But I think they just hit the sonication perfectly. So I don't know what it is. Conceptually, it makes a lot of sense, but I don't think you're ever going to eliminate the variation in the antibody step and all that. So okay, this is where we were comparing the differences. The key thing, the key lesson that we got here is that depending on how you normalize, you can get two different biological results. That's mostly what's in here. In one, there's a loss of binding in the resistant condition, and in the other one, there's a reprogramming, which is, we talk about reprogramming when we don't know what else to say. Right? Things changed. It was reprogrammed, but a systematic loss or a systematic gain. And this shows the difference between mostly loss and a little bit of gain, and pretty evenly balanced between gain and loss. If you look in the vignette, it makes it clear. I'm going to show you, I'm going to put up a sneak peek. What I'm working on right now for DiffBind is putting in these profile plots, actually using the profile plier tool that Thomas Carroll wrote. And there's now kind of one step, just like I said, DBA plot command, you can say DBA plot profile and get some pretty sophisticated plots. And if you look at this carefully, I'm also building in some annotations. So basically, I think the concept of DiffBind is much more to... I remember having a discussion with Martin Morgan 9 years ago, and he was trying to understand what it does, because it's not a statistical method. It's a tool that wraps the very fine statistical work done in other packages. But the idea is it collects your experiment and lets you think about your experiment as an analyst and deploy all these tools in as straightforward and consistent a way as possible. And at some point, he suddenly went, "Oh, I get it. It doesn't do anything." And I said, "Yes, exactly. You nailed it." And that's still the case. It's 15,000 lines of R, but it doesn't do anything. So it doesn't do anything that you can't do yourself if you have a very good command of R and probably about 14 underlying packages at this moment. But if you're just trying to analyze a CHIP or ATAC experiment in the most straightforward way possible, this is it. The last thing I want to say is come back to Seesaw, Aaron Lund's package, which does the windowing. I learned a tremendous amount from Aaron. He used to sit a few feet away from me in Cambridge. I learned a lot from his vignette that he wrote for the Seesaw package, and particularly about normalization. And I would encourage anyone who's interested in analyzing this to read that vignette, and it will make you a better analyst of DNA enrichment data, whether or not you use diffbind or Seesaw. All right, thank you.
So thanks a lot, Rory. Yeah, I guess we've actually finished bang on time, which is perfect. Thanks very much, Rory, and thank you everybody that's attended.